So hi and welcome to the latest speaker spotlight in the run up to our Immuno-Oncology conference. Today I'm joined by Chris Helsen, who's Director R&D and Head of Platform Development at Triumvir. Hi Chris, thanks so much for joining us.
Hi, thank you so much for giving me the opportunity. Pleasure
Great,
to be
well, great, thank you. Well, to start off with, could you introduce yourself a little bit further?
Absolutely happy to. Now, as you already introduced me, earlier, my name is Christopher Helsen, Director R&D and Head of Platform Development here at Triumvir. And just to give a little bit background about Triumvir, we are really a company that's built around the idea of engineering T cells with the goal to enable them to identify cancer cells and then elicit a toxic response. And we have had really great success with being able to bring a couple of items into early clinical trials.
Great, and I believe you're one of the initial founders of Triumvir. So what would you describe then, I think you've touched on it, but what would you describe as Triumvir's scientific vision? Why was it that you wanted to found
Well,
the
I can say a little bit about my own vision, and that is really that I believe nature has optimized biological systems through millions of years of evolution. So I think that the most effective way to really harness a biological system's power is trying to do as little artificial engineering as possible and really try to work with established biological mechanisms. And right now, doing so with our technology, we have been able to really develop a system that has brought amazing results in preclinical development.
Great, perfect. And then to focus a little bit on your current role at Triumvir, what is it in particular that the work that you're leading for the company?
Okay, happy to. So as Director in R&D here at Triumvir, I have the pleasure to work with a really highly skilled and motivated team. And we're always trying to push the boundaries to get new development ideas into ultimately patients and clinical hands. And as such, we always need to develop new targets. We need to identify new receptor designs. And really, right now, it is exciting to then do all this work, evaluate them first in vitro and in vivo, and then ultimately bring it into clinician hands and hopefully to the benefits of patients. And right now, we're actually doing that with undisclosed targets. So we're looking forward to that.
Perfect. And I think, obviously, as opposed to some of the CAR and TCR approaches that are out there in the cell therapy space, Triumvir focuses more on the TAC hybrid molecule. Why is this approach advantageous in this space?
Good question. Now, to answer that, I'd like to first make really sure that we acknowledge how amazing the results of the CAR technology as well as the TCR approaches, especially the TIL approaches, have been. I mean, patients are alive today because of those approaches. So I think it's fair to say that these approaches have been a really game changer in the field. And our vision is really to build upon that, right? And if you think about the CAR approaches specifically, they have a great advantage in the sense that they can target any surface antigen that you can think of, but a little bit of a disadvantage in the sense that the way they activate the T cell is an elegant but yet artificial way of doing so. And the consequence of that is that you sometimes have overactivation, a very high cytokine production, which then does lead to toxicity that we see in the clinic that limits certain areas of use for that technology. On the flip side, if you look at TCR technologies, for example, TILs, they have shown very good results, even some success in solid tumors, which is really hard to get, but extremely hard to produce. And in many patients, you can't produce them because you can't isolate the right cell population. Now, what the TAC receptor does, it actually really tries to marry both ideas, right? So on the one hand, you engineer a cell and can target any surface antigen you like, and you're not dependent on any cofactors like an MHC, portraying an antigen as a TCR is, yet signaling is completely through the TCR receptor itself, which means you do completely natural signaling. And as I said earlier, this approach has really allowed us to, in preclinical models for both liquid and solid tumors, show tremendous success where we really recapitulated what this idea has. You're very effective, yet you don't have this exuberant production of cytokines, as an example.
Great, no, that's really helpful, thank you. And I mean, a lot of the success in the cell therapy space has been more in the hematologic cancers, so cancers like leukemia and areas like that, with solid tumors proving more difficult in general. What are the main challenges of trying to target the solid tumors with cell therapies?
Well, That's really the holy grail question, right? So if you think about it, the solid tumor is really a complex question. And one could argue that liquid tumors have just less of that environment and might have less of those complications. And that might want to be one of the reasons that so far the success has been very focused on the liquid tumors and more challenging in the solid tumors. And if you think about a solid tumor environment, we need to just acknowledge how really difficult and complex that environment is. We're talking here about hypoxia, we're talking about acidic environment, we're talking about nutrient deprivation, on top of which we are adding immunosuppressive environments like Tregs, checkpoint receptors, we have suppressive macrophages. So you really have any T cell or immune therapy really has its work cut out to combat that. So at Triumvira, really believe there's a first really good step is to have an optimized T cell to really be able to combat that. And then further down the road, we believe we can probably further enhance this effectiveness via combinational approaches using other therapies that are already used, for example, checkpoint, to have a really easy example.
No, great. And obviously this is a really exciting space to work in, but what keeps you so passionate and optimistic about working in this field?
Well, if you think about it, I have the ability and the privilege to work with such an exciting team and cutting edge technology with the hope of bringing change to patients and improve clinical outcomes. And that is a really exciting proposition if you think about that. You're here, you're working, and you're working not on something esoteric, but you're trying to really bring change. And that, I think, is really what is so exciting.
No, for sure. And so to turn to the event, obviously you're speaking at our Immuno-Oncology week-long online event in July. What are you most looking forward to about attending the conference?
I have to admit that I really enjoy listening to other speakers and their brilliant presentations, because it's always nice to know what other people are doing in the field, what their ideas are, because we don't exist in a vacuum and many good ideas exist. And in conferences like those are very good to soak in some of those ideas and maybe exchange some general thoughts about the topics.
Perfect. And what topic are you going to be addressing yourself in your presentation?
Well, I will be looking forward to talk about our clinical development of our current TACO1 HER2 agent that is going into clinical trials phase 1 as we speak. And I will be talking a little bit about the preclinical work that led to the clearance of the IND.
Perfect. And then the final question then is, what are you most looking forward to doing after the pandemic is over?
To be fair, to actually attend these conferences in person, because nothing beats personal networking, to be honest.
Yeah, no, I think it'll be excellent when there's opportunities to do both the online and then also the in-person catch-ups as well. I think a lot of people are looking forward to that for sure. Well, thank you so much for taking the time to talk to me. And as mentioned, Chris is speaking at our Immuno Week online, so you can catch his presentation there. Thank you, Chris.
Thank you so much, and you have a great day.
Thank you.